All right, so let's talk a little bit today about diagnosing infections, chapter 15 in your text. So, what causes a condition? There are several ways that you can use to identify bacteria in a patient's specimens or in samples from nature. One method would be the phenotypic type of method. These types will consider both the macroscopic and microscopic morphology, physiology, and biochemistry of the organism. It'll look at colonial formation, color, the different types of nutrients they can use, and how they react to different biochemical tests. Serological analysis, we can look at their immunology, what type of antibodies do they form. Genotypic analyses, we're looking at the genetic techniques that increase. Uh, these techniques are increasingly being used as the sole source for identifying bacteria. One of the reasons we go with the genetic is they're going to tell you pretty much down to strain what bacteria you're dealing with. And also, in many cases, these types of tests are much quicker. But we would use data from a cross section of these methods can provide a unique profile for any bacterium. In the old days, you would do a combination of these to determine your type, and then in the early days of genotyping, a combination of all three of those was developed. So again, just another way of showing each one of these different types of tests. Again, in the phenotypic test, you will look at the microbes, macro and micro morphology. Again, what does the colony look like? Do they grow in individuals? Do the colonies merge? Do they produce yellow colonies, or the colonies that are cream colored? All of those different tests. Genetic means of identification are being used as a sole resource for identifying bacteria more and more and more. This increases in point of care diagnoses. The test can be performed bedside. Samples do not have to be sent out to a lab, then multiple test runs, and patient care can continue without delay. Some other techniques, MRI, computerized tomography, and positron emission tomography or a PET scan. We've all heard of PET scans, we've all heard of CTs, and we've all heard of MRIs. Uh, in phenotypic methods, we're going to look at the traits that an organism expresses. Uh, what it also is going to involve examining microbe appearance and behavior. Again, as I mentioned before, the different types of enzymatic activities it can carry out, the physical conditions it thrives in. Does it grow better at 37 degrees or 25 degrees? What antibiotics does it happen to be susceptible to? And the chemical compositions of its walls and or membranes. These are the results you get from things like the gram stain and the capsule stain and some of those other stains we did in lab. Antibodies response is exploited for diagnostic purposes when a patient sample is tested, looking for specific antibodies to a suspected pathogen. This is kind of the immunological test is what we'll talk about later on in the ELISA test is an antibody test. Oftentimes, these types of tests are easier than testing for the microbe itself. In these types of tests, you're not actually looking for the microbe itself. You are looking for the body's or the cell's response to that microbe. Laboratory kits for these types of tests are available for immediate identification for a number of pathogens. Possibly you've been to a clinic and had what was referred to as a rapid strep test done. A rapid strep test is an example of an immunological test. Genotypic methods have advantages over phenotypic methods in that culturing is not always necessary. In phenotypic studies, you always have to grow the bacteria, you have to grow up colonies of it. In these genotypic methods, many times you do not. Again, these genotypic methods are increasingly becoming automated, whereby you get very rapid results. and these results are often more precise than with phenotypic methods. The one drawback to phenotypic methods is they can tell you have an E. coli infection. They're not necessarily going to tell you that it is a particular strain of E. coli. Some E. coli are very virulent and can cause lots of pathogenic infections. Other strains of E. coli are not so serious. A viable non-culturable bacteria, these are things that can't be grown in the lab. Therefore, their identification is pretty much limited by genotypic methods. If you can't grow it in the lab, you can't perform the phenotypic methods. Success of identification and treatment oftentimes will depend on how the specimens are collected, handled, stored, and cultured. For these phenotypic methods, aseptic technique is imperative. You do not know and cannot tell what bacteria or microbe you are dealing with if your sample has become contaminated. So it is imperative that aseptic technique be followed. It is also imperative that it's sterile, that your sample containers and other tools be sterile. They can't be contaminated from the environment or the patient. So again, these are some limitations of the phenotypic test. Care should always be taken when samples are collected that may contain resident microbiota. If you're taking a sample from a patient and you're taking, say, a skin scraping or a fecal sample, all of those things are going to have resident microbiota associated with them. You should only be sampling the infection site, and be sure to steer clear of any surrounding areas, because the more the surrounding areas you sample, the greater the possibility of getting those resident microflora or resident microbiota included in your sample. Some sampling sites that you may encounter: from the mouth, you can get saliva and sputum. From other areas, you can get sweat. You can get a blood sample. You can get a urine sample. You can take a sample from either pass through the ureter, or through the urethra, or you can collect it from a catheter. You can get a punch biopsy of a section of skin. You can take a skin sample, vaginal swabs. You can take fecal matter, spinal tap, surface of the skin. All of those things can be used. This is one type of collection apparatus. This is a culturette swab. Has a sterile swab on one end. Has a chamber containing a growth medium. Many times these things will also contain a preservative or a transport medium. You've got a long swab. Again, it is in a plastic case, and a tamper evident seal. That way, once you collected your sample, the lab will then know whether or not this seal has been contaminated or messed with in any way. Sputum and saliva. These are saliva samples. They're obtained by having the patient either spit or drool into a container. If you've ever done the 23andMe or any of those types of home DNA tests where you can find out your heritage, many of those will contain a sterile tube that you are told to spit in. It'll have a line on the bottom of the tube, and it'll say to fill it to that point with spit. But that would be a saliva sample. Sputum is discharged by coughing or collecting with a catheter. Saliva is going to be stuff that's in your mouth, whereas sputum is going to tend to be more down in your lungs, in your trachea, etc. Contamination with saliva should be avoided since it contains millions of bacteria per milliliter. One of the problems with getting things coughed up is they're going to many times have a saliva contamination. So reaching down and collecting it with a catheter is a much more efficient way of ensuring your sputum samples are not contaminated with saliva. Urine samples can be taken aseptically from the bladder with a catheter. Two different types of collecting urine from a urine stream coming from the urethra. You can have what is called a clean catch or a dirty catch. A clean catch is taken by washing the external urethra and collecting the urine midstream. In other words, you start, go for a few moments, and then take your collection. Any contaminating microbes from the clean catch can be differentiated from pathogens. A dirty catch is where you take the first voided urine, and a dirty catch is required for some diagnostic techniques. When you go in and take a drug test, for instance, for trying to get a new job or something, many times they will tell you whether or not they need a clean catch or a dirty catch. The mucus lining of the urethra, the vagina, and the cervix can be sampled with a swab or an applicator stick if that is the type of collection that you need to acquire. Skin samples can either be swabbed or scraped with a scalpel to expose deeper layers. Wounds are generally going to be sampled by swabbing or by a punch biopsy tool. Fluids such as blood, cerebrospinal fluid, and tissue fluids must be taken by what is referred to as a sterile needle aspiration. And antisepsis of the puncture site is extremely important. Again, you want to make certain that as you are inserting the sterile needle, you are not picking up bacteria and contaminants as you are going in. Therefore, the puncture site must be cleaned. Many times you will see in folks that aren't allergic to iodine, a large betadine wash or an alcohol wash will be placed on before those needle punctures are done. Some other collections or specimen collection techniques. You can get samples from the eye, you can get things from the ear canal, synovial fluids in your joints, nasal cavities, by a swab in the nasal cavity. Anybody who's had a COVID test over the last couple of years knows all about that. Diseased tissue can be surgically removed. That process is called a biopsy. In any of these cases, the proper collection, transport, and storage is essential, especially this end part down here. Most of you are probably in any field that you're going to go into, you're going to be collecting things in a somewhat sterile, clean environment. In a previous life, I did a lot of testing in the field where I was taking water samples and mud samples and all kinds of samples in nature. And at that point, these things were having to be transported. So I was collecting them maybe today and tomorrow, and they were going to be transported to the lab on Wednesday. It's Monday where I am right now. And so it's possible that you may collect things for a couple of days in certain instances. So it's very important that storage is essential. Some things need to be stored on ice. Other things would need to be stored you can store things at room temperature. It all depends on what type of test you're going to be doing and what type of test what type of organism you're testing for. Some things can be stored perfectly well at room temperature. Other things you had to be testing or storing them on ice. When I was doing my work, in another life, I would go and take samples, and I would be staying in a hotel. I would stay for 3 or 4 days in an area taking samples, and I would collect all my samples. I would keep them in the mini fridge in the hotel. Don't tell the hotels I was doing this, they might not be happy about it. But I would keep them in the mini fridges in the hotel, and then the last day I was there, I would make all my collections. I would put one of these those cold packs that you can buy. You break the chemicals, you shake them up, and for the next 12 or 15 hours, they're nice and cold. I would get one of those, put that in my shipping box, and then I would ship them off to the lab on overnight mail, and they would be there in plenty of time, still be cold and still be viable. So I was worrying not only about collection, but I was worried about storage and transport. Here's an overview of different lab techniques. All of these are the different lab tests that can be done, all the different results. This is This is the thing I would fill out. I would say over here, I need this test and this test, this test, this test, and this test. And I would say, where the test came from. Mine was a little bit different because this is for human beings. Mine was for environmental samples. But you have the same kind of thing. This is a throat culture. I need this test, this test, this test, and this test. And then the reporting lab then would fill in their results down here. Different ways you can directly observe your organisms microscopically. You can do that with fresh or stained specimens. This is one of the most rapid methods of determining presumptive and sometimes confirmatory microbial characteristics. It may only mean that we need to know the difference between is it gram positive or is it gram negative. If that's all we need to know, give me 10 minutes and I can tell you. Is it an acid fast or positive? Again, give me 10 minutes and I'll tell you. Other types of things, however, may require longer. Selective and differential growth. We talked about that in lab early in the semester. A specialized media is one that is used to enrich a pathogen present in small numbers or that may be easily overgrown. Two different types of media. We had selective media. This is used for non-sterile specimens containing a diversity of bacterial species. It's going to encourage the growth only of a suspected pathogen. So you've got a whole bunch of stuff, you don't know what it is, but you have a particular type of media that only one type of thing will grow on. That will select. So if you're looking for you're only concerned about gram-positive organisms, you put it on a media that is selected for gram-positive, and if your organism is growing after a few hours of incubation, then you have the right type of media, you know what you're dealing with. A differential media is used to identify some characteristic or some fermentation patterns. A very classic differential media is a media called eosin methylene blue. We use it in our lab here at University of Arkansas Pulaski Technical College. We use EMB here, and EMB is a differential media that shows us a very characteristic type of pattern for fecal coliforms. Now, we've talked about fecal coliforms throughout the semester, and please remember that while E. coli is a fecal coliform, all fecal coliforms are not E. coli. And this media, any of the fecal coliform bacteria will grow on EMB media, and it will show a very distinct color pattern. There will be a very bright, shiny green color to the colonies that grow, or that colonies that are produced by fecal coliforms on EMB media. That green color is the differential part. Here's a flow chart or a dichotomous key showing how to separate genera of various cocci bacteria. So basically, what has happened here, somebody took a sample, they looked at it under a microscope, and they said, oh, these are cocci. The cells are of a cocci shape. How do we adjust my camera here because I'm not getting the bottom of the slide. I apologize. So the first thing we do is we do a gram stain. Well, we have a gram positive and a gram negative. The next thing we did was we checked for oxidase and catalase activity. We checked the gram positive one. Is it catalase positive or is it catalase negative? If it was catalase, if you have a cocci that's gram positive and is catalase negative that is growing in chains, you have a Streptococcus organism. If, however, it's catalase positive, you then have to determine, is it an aerobe or is it an anaerobe? If it is an aerobe, then it is a Micrococcus bacteria. And if it's an anaerobe, it is a Staphylococcus or Planococcus bacteria. The gram-negative ones, we're going to look, are they aerobic or anaerobic? if they're aerobic, are they oxidase positive or catalase positive? This is this group of bacteria, the Neisseria, etc. And Bolonella is an anaerobe that is oxidase and catalase negative. So this is the way we would use these multiple different tests to determine what a particular bacteria is. And this type of thing where we go, it's a cocci, is it gram-positive or gram-negative? If it's gram-positive, is it catalase positive or catalase negative? If it's catalase positive, is it a strict anaerobe or is it a facultative anaerobe? This is referred to as a dichotomous key. Dichotomous means two questions. Each of these pairs is referred to as a dicot. Some biochemical tests that we may do. These are where we're looking at the physiological reactions of certain bacteria to different nutrients and other substrate materials. These tests will provide evidence of enzyme systems present in the species. If it can metabolize glucose, for instance, then it contains a certain enzyme. If it cannot metabolize glucose, then it is lacking in that enzyme. These tests are based on enzymatically mediated metabolic reactions, usually visualized by color change. The microbe is going to be cultured in a medium with a special substrate, then tested for a particular end product. That particular end product will produce a color change or a color variation. Microbial expression of the enzyme is made visible by a colored dye, and no colorization or no change in the color means that it lacks the enzyme from utilizing that particular substrate. And again, here we have an unknown microbe in my substrate, the enzyme present in the microbe, so we get a color change. Here, color doesn't change for a negative result. Right. This is a rapid chemical test. This is a rapid biochemical test where agents have been placed in all of these little wells. You put a small sample of your bacterial culture growing in each one, and you look at different results as you check these, 49 different tests that are available. This is called an API strip. This is a fairly old, this is one that's going to identify lactobacillus organisms. We're going to stop right there for a few minutes, give my throat a rest, and I will be back to complete this chapter.